Hello and welcome to Bite Size Biocompatibility episode 10. 10, the big 10. And we're going to be discussing all about local effects, aka the implantation study.
Yes, and we're so excited to be able to discuss and go into what is one of areas I'm very passionate about and involved with. So we're going to go through the nitty gritty here to be able to try to make this rather seemingly simplistic aspect that evolves to extreme complexities into the most digestible
format possible. So we do cover quite a lot with it as well, but hopefully it gives you a little bit more of an idea about how to approach an implantation study, where it can be used, the power in it, but also where it shouldn't be used.
And a warning to those who are a little squeamish that we will be discussing obviously in vivo, so therefore animal testing. So please be aware and conscious that if this is something that is impactful to you, that is taken into account when proceeding with this episodes. But otherwise, we look forward to the discussion and the topic. So look forward to then.
See you then. Hello and welcome to Bite-Sized Biocompatibility, the educational series focused on making the technical content of medical device biocompatibility digestible. My name is Leah Williams.
Hi, and I'm Beau Rollins with Intrinsic Medical Group, and we're excited to take you along on this journey.
All right, hello and welcome back to Bite-Sized Biocompatibility. Today we're going to be talking about local effects. Also,
Episode 10.
Episode 10, yeah.
I know.
The big 10. We made it. Yeah, local effects are also often synonymously referred to as an implantation study, but actually first I want to know, is this always accurate? Is a local effects study always an implantation study?
I mean, I would say there's never an always with anything that we do in biocompatibility.
But most of the time you are looking at implanting.
Yeah, when people usually ask, like, how come we can't use TRA to resolve our risks and our effects regarding implantations, because they're not analyzing the same thing, right? And implantation is looking at the local effects, what is happening around the implant itself, around the device, and those specific interactions there only. And that's really where the focus of that study is, where when we talk about toxicity and systemic toxicity and the TRAs and all that, is looking more across like a systemic interaction with complex systems and organs and everything else. So
like metabolic pathways.
There's a very distinct difference, and it's one of the reasons why, at least for now, we don't have an alternative solution for implantation. I think this is probably a good time as well to throw the little caution out there to people who have tuned into this, that here we will be discussing and reviewing implantation of medical devices, which currently is on animals. So the discussions today will be focused around these types of studies, and just as a caution to those out there that
we're talking about animal testing today,
are sensitive in that we
be aware.
Be aware it is an aspect of medical device that is not near with an alternative solution. And so what our focus is on here, and what every biocompatibility specialist focus should be when they go to this, is that they conduct the most meaningful and appropriate test to evaluate the risks and harms of their device.
Yeah, absolutely. You've said that beautifully. Really make sure that if we do need to perform a study like this to demonstrate that the device is safe, making sure that the data that we're getting out of it really does answer those questions and fills in those risk mitigation gaps that we have from our biological evaluation.
Yep. So I'm excited to talk about this, because this is one of my passions. This is one of my areas of focus, right? So I'm the U.S. co-chair for implantation. I have an amazing partner, Chris Parker, who's also the ISO convener. So shout out to Chris. We do everything we can with this to make sure that we are approaching this to get the most appropriate data possible out of there, to really be able to utilize it to its maximum, understanding that there are things that we need to verify. The last thing we want anyone in our industry wants is for us to have a device used on a human and have complications or results with that patient, adverse results, right, that were unexpected. And the fact that that happened is because we missed it in our evaluation testing.
And I think this is actually a really great point where we can really highlight the power of this test as well, because even though it is an animal study, there is a lot of very valid clinical data that you can get from this.
Yes.
So
no, I think that's a great way of putting it, because if you look at -6 an implantation, it's a relatively straightforward, somewhat simple looking type standard. And the reason behind that is there's actually a lot of flexibility and variability that we allow within this. So it's simple because we list the basic requirements to meet an implantation study, but we leave a lot open to customization. And that's intentional to be able to allow you to design this in a way that gets the most relevant data. And we want to continue this on. If you look at the most recent revision that has been released on, which just came out, we added into there a new annex for neuro implants, right? And we're moving more and more with these annexes to be able to provide specific guidance for very specific custom implants. And we want to continue that approach. So that's a future aspiration, is to continue to give these types of guidances for these areas. But it's just to further show that you can do almost anything with this type of study. And it's one of the reasons why, unfortunately, it also gets confused.
Yeah.
So one of the things that I like to highlight a lot and that we see often is that the implantation, the -6, is confused and used to mitigate systemic toxicity.
Right. And it's actually in the title of -6 that this is to evaluate local effects and not systemic effects. And I think a reason why that gets confused with the systemic toxicity is that you think, well, I think there's actually two reasons. You're implanting a material or a representative portion of your medical device into an animal, so therefore the whole system is being exposed to that. And then also the other side of it is that, as we mentioned in the last episode on systemic toxicity, you can tailor your methodology to look for both of these biological effects within one study as well. But with this, and Beau, you're going to have to talk a little bit more on this as well, you have to be very careful about how you actually set up that study to make sure that you're meeting the requirements in terms of number of animals, in terms of what results you're looking for, what data that you're looking for as well, to meet the requirements of both standards. Don't think that you can just do an implantation study and that it will automatically meet those requirements for both systemic toxicity and local effects.
Yep. So the first step on this we talk about many times here is local effects. So this is about how tissues and the body interact with the physical structures and materials and whatever residuals or coatings may be on there. So it is very much a focus on that specifically. That inherently then means that we have both, good things about this and complications about this. Too many people will sit there and try to look at the local effects, and as Leah's discussing that about trying to bridge that into other aspects and concerns, we see this apply incorrectly for a lot of devices that aren't implants. So the premise behind this, and that's why it's called implantation as well, is that this was designed and developed for devices intended to be implanted.
Right.
Not necessarily a permanent implant. You might have an implant that's put in for 2 weeks or 4 weeks to do some sort of action, or as the body heals and then removed. So you still need to look at those effects. But the problem we see often is that the way that the table was designed is that implantation was listed for any sort of cumulative long-term device.
Right. So I guess this leads to the question of, before we jump into looking at how maybe one would approach doing an implantation study, when is it not appropriate to be used? And it sounds like, from what you're saying, from the tones there, it sounds like you have a couple of examples in mind.
The one I like to use the most, because it's so frustrating, is you have an intermittent urinary catheter,
right,
the device has about 5 minutes or less, actually more like 2 minutes, contact with intact mucosal membrane.
Well, think about how long it takes to go to the
restroom, to empty your bladder. So you're talking a very short duration of contact, right? And yes, you do use the bathroom more than once a day. So over count of a day. If we want to say like clinically wise, we usually look at 6 times as being the amount you would catheterize. 6 2-minute periods, right? Even if we want to do 5 minutes there. So we were talking about 30 minutes of contact time, but it's never even continuous in that. But because you do this every day, it falls into that cumulative long term. And I have had regulatory agency after regulatory agency ask for an implantation of an intermittent urinary catheter to be able to evaluate, because it's a long-term device through cumulative use.
Right, and it's really important to note that for those maybe less familiar with like how catheters are used, the worst case contact classification actually is intact mucosal membrane. It is not implanted. It does not go subcutaneously or within any tissues. It is in contact with an intact mucosal membrane.
That is correct. And there's no time allowed for any sort of cellular stabilization around the device. But the model, when you look at this and what they ask for, is basically having sections of the catheter cut and implanted into the backside of like a rabbit or even a rat, and looking then at how that works over you have to go longer than 2 weeks. So they want to look at least a 2 weeks. Some have requested 4 weeks.
Yeah.
And how does this have any relevancy whatsoever to the use of this device?
It's not clinically relevant at all. And actually you hinted at something there as well, that you have to go longer than 2 weeks. And you mentioned the tissue stabilization as well. And I think that's one of the key defining parts about doing any sort of implantation or local effects study, is that it needs to be an objective of the study is to have that tissue stabilization around the device, so you can have a look and see how the tissues respond to the device being in contact once they have stabilized. So therefore, any device which clinically isn't intended to be used in a way where it allows for tissue stabilization of the device around it, an implantation study just isn't relevant there.
So when you talk about implantation, one of the setups and designs is the length, right? How long are you going to do an implantation study for? And that should reflect the clinical use of your device. When we're talking like a permanent implant, you're not going to go like years in an implantation study. We generally, for like the United States and CE regulatory markets, you look at around 13-week to replicate a long-term.
And from my understanding, that's scaled from a human lifespan down to like the animal model lifespan, right?
Correct. That was the initial premise behind it. When we look at Asian markets, specifically Japan, they request a 26-week. That is just because they want longer time for that interaction between the materials and the animal, which is fine if that's what they ultimately kind of want. But the reality is as long as your device is not degrading, 13 weeks is sufficiently long for complete stabilization.
Right. I guess the question that comes from that I have for you there is, it's also another thing of what the implantation study is not designed to do. Once we've hit that 13-week mark, there aren't other biological effects that we can maybe mitigate with that, say looking for screening for like cancer risk or anything like that, right?
I mean, if you, not generally speaking, because the intent is look at local, but obviously like if you do implant a device and you went for the long duration of the animal's life, and you noticed cancerous tissues upon necropsy in that, then obviously you've potentially got that and you need to investigate further. So that would be something that you could indirectly look for, but the intent is not to be able to look. And part of that is also the dosing aspect. So you'd have to have something very mutagenic, that you're implanting from that initial.
Right. And what we've discussed previously is that manufacturers don't intentionally put these things within their devices. So.
Correct. So that should not be an outcome. These are always the types of studies that, no observation should be able to mean that you can leverage that to say, hey, if we didn't see toxicity in the animal, then that means that it doesn't cause toxicity.
No.
But it's not designed to evaluate that. So even though you can make inference to it, and sometimes you can leverage these as a supplement data point, it's not a conclusive data point.
Right. I guess like my related question to that then is, why is there any point in going beyond 13 weeks? Is there any data that you might be able to get from that, or what situations maybe would you want to consider looking beyond 13 weeks?
Beyond 13 weeks, I'd personally consider, if I don't know the degradation aspect of my materials, or if I suspect that there will be degradation of my materials,
and it's a slowly degrading.
And it's a slow degrading. If I know like I have radiation sterilization that's done on my device and it causes some sort of degradation profile over time, then perhaps a longer period, especially if I have other data to show that after, a year or something that we start running into issues. Now, setting up that type of study would be a lot more complicated and expensive. You could even look at maybe aging samples, and we can talk a little bit about that later in sterilization stuff and that, but you can custom design these for anything that you really want. The reality is, like, do you need to? Yeah. And that's where you have to ask that up front. So if you have inclinations of potential risk in that, you can design this with absorbables as well. So if you have an absorbable device and you're going to do an implantation study, you obviously need a time point that's around implantation to make sure there's nothing right after. But then you also want to set up your study to whether you either accelerate that degradation aspect, if it's hydrolyzing of bonds of the material and that, so that way when it's put into, it expedites that degrading and absorption aspect of the study, because otherwise you will be looking at doing, and I have done these as well with canines and porcine models, where you do like 2-year implantation study, because it takes 2 years for that absorbable to fully absorb, and we have to monitor and observe what happens through that.
Right. So we've kind of danced around it a little bit, but maybe just in layman's terms, if you had determined that it was clinically accurate and appropriate to perform an implantation study with your device, how would one approach that? What sort of things would you look for?
Very, very good question, and a good high-level question to step back on, because I know I dive down into the too many details here.
All good content.
So when we step into an implantation, first off is, how is my device going to be used and exposed to the patient? So that's going to then mean that I need to expose it to that same type of situation, right? So if we need muscular, bone, subcutaneous tissues, that will depend then on where you're going to put the implant, and that will also generally lead to what model you want to use. Most implantation studies, I would recommend using a rabbit. And then if you need to go like for bones and stuff, you can do rabbit, but then sometimes it's better to be more into a sheep thing. So
like an orthopedic device, for
location will impact that a lot, as well as life span of the animal and the duration of your study. So if you need something really long term, you're not going to be able to use like a rabbit or whatever. They won't
You
live
need
long
something
enough.
that has a longer lifespan.
That has a longer lifespan to be able to compass that. So these are factors you start playing into. The other thing is, you never implant your actual device, because our devices are designed for humans, which are much larger than the animals.
Right.
So you're going to have to be able to create a custom implant. A lot of them are by the standard shaped like a pill or like a long fragment or whatever that you can put in.
And this actually relates right back to Dash 12 as well, in preparing for that biological endpoint testing and making sure that that sample really is representative of your final finished medical device, in terms of it's got all of the constituent materials that would contact the tissues. It's representative of the manufacturing and the finishing processes as well, as any sterilization that might have been performed on
it. And honestly, that's the hardest part of these studies half the time, is being able to create the samples to implant that replicate the normal product.
And I guess that a roadblock that I see with using that pills type study as well is that there's a lot of devices out there that say maybe have some sort of porous coating on it or some sort of surface finish that actually maybe you also want to have a look and see. It's specifically been designed to elicit some sort of beneficial biological response. So you want to be able to replicate that as well within your study.
Yep. So those are factors you have to start including. We've mentioned about stability. So there used to be a little bit more prominent in the previous revision. It's been dumbed down a little bit, or softened. Maybe that's the better word. Softened a little
Politically correct there.
Yeah. Where we would note that you need to wait ideally 2 weeks, because with any surgery and any aspect we're cutting into the animal, right? Especially if you're going into bone, you're drilling into bone and things, you need to give the animal time to heal around from that surgery and to then allow the tissues and that to interact with the material. So,
and even if those surgical wounds are going to be clean cuts based on the methodology of how they're generated, it still does take that time for it to heal.
Every animal model heals differently, so we didn't want to try to go through and list out for each model what is its ideal heal rate. 2 weeks is a safe bet for all animal models. So generally speaking, don't do anything less than 2 weeks, because this whole test isn't a pass-fail. That's the other thing to keep in count. you don't have an acceptance criteria like other tests where we're looking specifically for like a certain type of response. We're comparing everything back to a control on this test. So if you are looking at 1 week, your control might have healed faster than what your test article has. You can get different healing rates. And so then now you're trying to compare, and you have inflammation seen regardless.
Yeah.
The general control that's used for this test is ultra high molecular weight polyethylene. It's very smooth. And it's very thin. So the slit needed to place this in generally is very tiny. And so the healing around the control happens relatively quickly. But if you have like especially something with porous coatings and stuff like that, and you have to create a larger slit to fit your sample in, your healing is going to take longer.
Right. And on that control piece, actually, it is worth noting as well that you don't have to use that ultra high molecular weight polyethylene. You can use a different control piece that actually maybe is representative of a previously approved predicate device or a material which is with known biocompatibility that is more similar to the materials you use. So I think actually this is a really powerful part that is very overlooked within these studies, is using the right control material instead of just defaulting to ultra high molecular weight polyethylene, because it might not be comparable at all to your device.
And the reason we use it, or it's listed, is because there is, you can buy this from a standards organization. It's sterile, it's already cut. So it's easy for the labs to use, and it's very inert and well characterized, but it very rarely is a representative control, especially when you're comparing local effects to whatever you're implanting. So from my experience, 9 times out of 10, it's not the best representative material to be able to control or compare against. That was one of the disagreements on the recent one is do we even list this as a
the recent standard update?
Yeah. Or if we remove it entirely and lean more to a predicate approach, which is more my preference and view, is that there's always some sort of predicate device out there. It may not be exact, but there's other things out there that are more appropriate in comparison to your device. Maybe not in same intended use. And a good example is on some of these is that we've run into is like, if you have a type of suture, and maybe your suture material is a new novel one that has some sort of elasticity aspect to it, or even absorbable aspect to it. There's going to be another material out there that's similar in porosity and flexibility that you can put in that will have similar effects with that cell interaction that yours will. And that's the important aspect that you need to focus on, because your entire results are going to be comparative. And having appropriate predicate is the most important aspect when it comes to setting up these tests.
My next question for you is, what about dosing? How do we know that we are appropriately dosing correctly with our implant?
So when we talk about dosing, that's where this is not a systemic toxicity test. So dosing actually has no relevancy to this whatsoever.
I'm so glad that you highlighted that.
Yes, we are not looking at dosing. We're not looking, and that's why this doesn't work for toxicity out of the gate. You have to customize that into this type of study if you're going to need and want dosing. Otherwise, we have number of sites. So we need 10 test samples and then 10 predicate sites. And those can be done in the same animal. And that's the thing where like we try to follow -2 with these. So a very simple muscular implant study on a rabbit, you're going to have 10 sites on one side of the spine that's going to be test, and then 10 of the control. And we get as much data out of that as we can. But keep in mind now that, we obviously have mixed things. So if you see things in organs and stuff, you don't know if it's control, if it's sample, or anything like that. And that's where ideally, like sometimes it's not even worth looking at the histopathology aspect of these, because we just want to look at those localized effects.
Right. I think that is such a key point. If you take nothing else away from this episode, take it away that you cannot just satisfy your systemic toxicity effect or effect with the localized effects endpoint as well with an implantation study.
And that goes back to my frustration on like an intermittent catheter, where the amounts that you put in to satisfy 10, and then generally speaking are going to be about 2 centimeters in length, right? When we equate that to dosing, if we were trying to, you are substantially dosing
you're
the animal to what a patient would ever see. So it really is just poor science, for lack of better terms, by trying to utilize this type of thing for the wrong products.
So we've talked a little bit more about how how you can't, or you can use it for systemic toxicity, but you really have to set up your study design, and typically your more standard approach to it isn't relevant to systemic toxicity. We've talked a little bit about how you'd approach doing an implantation test, the power of it. Now I want to go into a little bit about the results, and I want to start off with irritation, because that's something that you do see in a test report, that irritation is reported. And how does that relate back to the irritation biocompatibility effect that we have to evaluate for, one of the big three,
so
if at all?
No, it's a good point. So the way that these are read primarily is on an irritant score. And so when we talk about, even with implantation, we'll have clinical observations that we go through, and then we do with the necropsy, we'll be doing histopathology at all the sites. So we prep a bunch of slides, and we're going to be looking at those, characterizing the responses as far as being irritating, because we're looking for vacuolization there. We're looking for increased white blood cells and that to the area. All those are signs of systemic response that gets defined as irritancy. So it does cause confusion, because when you get your report back from implantation, it may be a slight irritant as compared to the control, or a moderate irritant. And most people's minds will initially go that, oh, I have an irritant here. Why didn't I see that in my irritation test? It's not the same, right? And hopefully through this kind of our previous episode on irritation, and we're talking about what affects and what drives those types of redness and swelling that we see on the surface of the skin, versus here when we're looking at influx of white blood cells, vacuolization around the material itself, or separation between the material. We do have cellular irritation that we can see through redness and swelling as well, through some of the dye stains and things. So there's a lot that can factor in, but it is historically been reported out after everything's surmised, whether or not it's more irritating to the local tissues or less irritating by all these complicated responses.
Right. So the takeaway there is -6 does not cover
-23. Yes, it does not. Unfortunately, you can't leverage it to look at that, even though it does talk about irritancy in the result.
Yeah. Just be aware of that key difference there for sure.
And it's definitely one of those too, when you're talking about slight irritation in comparison to the control or the predicate, right? That's what type of result you're going to get on the report. And regulators are always going to be looking that there was no observed irritancy to the control. If you do get some, this is where it's very important then to start stepping through what was the differences seen and how is that clinically relevant, or is there a clinical risk to it? Because just because one of the things that we'll see a lot with, especially if you use a high density polyethylene, but you have like a suture, if you have something that's flexible, that moves inside the site versus something rigid, that movement actually causes the cells to have a bit of a irritancy response against the material.
Right.
Same thing if you have any sort of porosity, you'll get cellular ingrowth, which will equate to irritancy when compared. So identifying what those responses are that drove your slight irritancy is very important, and then determining whether or not that's a clinical risk or not. And honestly, this is the biggest challenge with those results, is having that discussion with a regulator in a way that they're comfortable and confident, because most of them just want to see that there was none.
Yeah. All right. I mean, this has been a fantastic discussion. Is there any other final points that you want to put in there that you're dying to say?
I mean, implantation does have a lot of power. I think we mentioned in the previous episode about wound healing studies, right, when we talked about systemic toxicity, and I mentioned that it's more in -6. So wound healing studies are built off a -6 premise. It's not about dosing the animal at all and looking for systemic. It is about healing. So instead of a defect where we put a device in and suture up and leave it there, we create a defect on the skin, and then we apply and administer the dressing as per intended use, but we are watching how it grows back as part of the study. And then at the end, we will take slides of the wound area to look at the new skin and how it healed in comparison to, other predicate devices, which are usually like a gauze, right? Unless you're able to use like another predicate device. So there is a lot of power here with the implantation. Please do use them to make them clinically relevant and appropriate, so that way your data is meaningful, because at the end, we want to make sure that, as a patient heals or has this in contact, that we're not going to be leaving debris and fragments and things that will later on either not be metabolized and stay. And that goes with the new -1 on bioaccumulation and what happens to things that are in the body. A lot of people like to think that if you have a foreign material from your device left there, that it just stays there. The body just vacuolizes it or observes it as a foreign material, but then leaves it alone.
That's definitely not the case.
It's not always the case.
It may be the case, but it's not always the case for sure.
Yep. And so these studies, you can get very complex where you can put labeling marking on your materials. So that way you can actually see how they migrate when they're metabolized, if they are metabolized or transported around the body.
Some sort of like chemical tagging or something.
Radioactive tagging, iodine tagging and stuff. Like there's a lot of complexities that you can put into these studies to really evaluate additional effects.
Yeah.
Or they can be super simplistic where I'm just creating a simple defect, shoving it in, and in 4 weeks I'm going to look at how it interacted with that.
Right. So a really versatile and powerful study if used in the right way, but definitely a few points there, especially with the systemic toxicity and not using it for irritation or the irritation biological effect as well. So yeah. Yeah.
And similar to like the systemic toxicity, it is very important that you are very open and communicating with your CRO.
Yeah.
Please make sure that, you're sitting and going through these, you're talking about all these aspects, that you go there to observe how it's
and that you
handled.
Understand what's going on, so you're able to then justify the decisions as well and communicate that to any regulators and make sure that if you understand the data, then it's also more meaningful to your biological evaluation.
Yeah. And many CROs are even willing to cut samples. So if you came back and were like, I can't mold this into that shape, they may be like, well, send us a device and we'll cut it.
They have specific machining tools. Like I know that when I first heard that, I thought, oh, they'll bring out some scissors or maybe they'll have like a hacksaw or something like that. But actually they do have, a number of them have specific machining tools that can cut through metal, ceramics. And
honestly, it's all over the board. And that's where you need to be involved to know, because some of them do just pull out scissors or tin snips, wipe a little alcohol on there, and will start cutting the sample in what they think is the most appropriate. You want to make sure that they're including all the materials, right? Because if you do have varying materials through the construct, you got to make sure it's representative, right?
Or even a gradient from like the different processing of the same material or anything like that.
And then if it is metal in there, then they're probably going to use some sort of bandsaw or a type of cutting.
Now we talk about
particulates, but also lubricates.
So coolants as well. So
like I said, this can turn a nightmarish very quickly. I have seen with like cobalt chrome where you couldn't get it machined into the appropriate size, and so we sent it to the lab to have them try to cut it, and then them using plasma cutters and other things to try to cut this, and then changing surface characteristics and or adding residuals. And then we need to sterilize it. There's a lot of things that can get complicated really quick or go wrong with this if you just blindly send and say, cut them up and figure it out.
Yeah. Well, it's been absolutely incredible to talk to you about all of this. I think we're right at time, so we'll leave it there for today. But yeah, hopefully that made the topic of local effects a bit more digestible. As always, feel free to reach out to myself or Beau with any questions. And yeah, see you next time.
See you then.